Hello, hello. Welcome. Welcome to this seventh edition of our Doctoral Derby finals. I'm very happy that you all gathered here in Pilar Box, and of course also for those who are joining online, a warm welcome to you all. What is this Doctoral Derby all about? Simply put, it's a public speaking contest among PhD and postdoctoral researchers, and here at the VUB. And simple is really the key here. These are all passionate researchers, right? They can talk for hours and hours about their research. But can they also talk about it in really simple terms to some stranger in the streets or during a family gathering? That is really the challenge here. And on top of that, they have to do it in 3 minutes' time. Challenging, but they can really do it. Okay, what do we have on the menu today? I'm your host. I'm Hannelore de Grande. I'm the coordinator of the Researcher Training and Development Office. And we started this in 2018 during 10 years anniversary of the doctoral schools, actually. And it has been grown ever since and became more popular. I will briefly introduce about how the trajectory has been going. And then I will ask every PhD candidate, one at a time, to come to this stage and present their pitch to you. After that, they will get some questions by our fabulous jury over there. And we do that 9 times in a row with some physical exercises in between. And then it will be up to you. We will have a little break. You will be able to vote for your favorites. And the jury will deliberate about everything that they saw and will also present their winner later on. Of course, at the end, we will all celebrate in drinks and fries. So stay until the end. Okay. So what have our contenders done the last months? They have done quite a trajectory so far. So they started off with a training on how to summarize their research in an accessible way. And they got a training from our own colleague Sven, who you will have seen walking around here today and we'll see later on as well. So they had the opportunity after that to make some more changes to their summary, and then they had to hand in their definitive text. After that, they could follow an short online training on how to present their research into a pitch. To prepare them for the pre-selections. So we organized pre-selections for each doctoral school. And so they really were contenders. There were contenders from disciplines that are quite close to them. After these pre-selections, also the jury and the audience selected the ones, their favorites, and they got to this finale. These finals, before they got here, they got a more intensive training by the Floor is Yours about really how to present with impact. And so make some more changes to their pitch after the pre-selections. And they had the opportunity to record their own video with science figured out, and this will be become available in the upcoming months. So this is, of course, a great extra for them as well. And now we arrived here in the finals, and of course we are looking forward to see them talk about their research, right? why do we do all this, or why would they want to put themselves in this trajectory with quite a lot of stress along the way sometimes? There are three great awards lying ahead of them. So we have the Best Pitch Awards that is being offered by the jury. You can vote as audience for the Audience Award. And then there was also a jury that selected the best research summary last week. So there are three prizes. Could be three different winners, but it's also perfectly possible that somebody gets both the award for best research summary as well as the best pitch. Let's see. What do they further gain, of course, by following this trajectory? It has been a moment where they, a whole trajectory where they could really, where they were being coached along the way. Several coaches came by, Sven, but also the floor is yours, who have a lot of experience with coaching researchers in really getting their message across. So this is really is really nice, of course, to have all these coaches around and to have really grown as a researcher, to really have sharpened those presentation skills and those writing skills along the way. Of course, they will also enter the Hall of Fame of our Doctoral Derby. And last but not least, they get 500 euros working credit that they can use for their further research. Now, I would like to present the jury to you. From left to right, we have Lauren Pauwels. She's also a colleague from the Researcher Training and Development Office, policy advisor there. We have Gert van der Steen, who is the director of the Doctoral Schools of Natural Science and Bioscience Engineering. We have Floor Keersmakers from the Research Outreach Office, who is a policy coordinator from the Citizen Science contact point. And we also have Arushi Karo among us, who has been winner of last year. So I was wondering, Arushi, how is it to be back here in Pilar Box?
It feels great to be here back, also on the other side of the table, instead there on the stage. I'm very excited to see all of your pitches, and yeah, let's see how it goes. Good luck.
Thanks a lot. And then we have Leo van Grunsven, who is the director of the Doctoral Schools of the Life Science and Medicine. And lastly, we have Marie-Laure van den Houten, who is actually replacing our director of the Doctoral Schools of Human Sciences, Karen Francois. Great that you found the time to be here. Okay, I think we have done a lot of introductions, and it is time for our first presenter. And we're starting off with a great name. So I would ask my namesake, Hannelore Gerard, to please come to the floor. You can already give her a round of applause, sure. Her research pitch will be about bioplastics, stretching towards sustainability. I would say, Hannelore, you're the first one up. Take your time. Don't forget to enjoy it. The 3 minutes will start when she starts talking. And don't forget to stay on stage for the questions afterwards. Give a warm applause.
Imagine this, our modern world, but without any plastics. Difficult, right? Because plastics are all around us, and they have become indispensable. Plastic packaging, for example, keeps our food safe and fresh. Unfortunately, there's a downside, a big one. The production of plastics makes a major contribution to global warming and is turning our oceans into a plastic soup. But luckily, there's hope. Have you ever heard of bioplastics? The bioplastics that I study have many advantages. From soil to ocean, these plastics break down naturally, leaving no traces behind. That's what we call biodegradability. And instead of using dirty fossil fuels, these plastics are made from renewable resources, like food waste or wastewater. But the most fascinating part, they're actually not made by us, they're made by bacteria. That's right, it's like having millions of tiny, tiny factories turning useless waste into valuable plastic materials. Unfortunately, these bioplastics are not yet widely used, and that's what we are trying to improve with our research. By cultivating bacteria under varying conditions, we produce bioplastics, like this one, with a different chemical composition. The goal is then to study how the chemical composition of this bioplastic will affect the material properties. For example, just by changing what we feed to the bacteria, we can produce bioplastics that are really rigid or super flexible. Amazing, right? But why does this actually matter? Well, only if we can closely resemble the properties of regular plastics with these bioplastics, only then will consumers and companies be convinced of this sustainable alternative. And that's actually what we are trying to do with our research. Plastics are all around us. We cannot escape them entirely, so we have to make them better. Made by bacteria and biodegradable, these plastics will lead to progress and not pollution.
Wow, well done, well done. I think it's always powerful when you do it without slides, so
Thank you.
amazing job. Jury, do you have questions for Hannelore? Yes, Leo.
Very interesting. How much bacteria do you need to make such a small piece of plastic, and how easy is it to scale this thing up?
So in our lab, I'm producing these plastics on a scale of, let's say, 1 gram. And I'm doing this in a volume of half a liter. So it's really small scale. And this, for example, this sheet is made from only 300 milligrams of plastic material. Luckily, we have a lot of instruments to test the properties of these plastics that don't require big sample sizes. But of course, scaling up is something that is not possible here in our lab at the VUB, but is done actually already at this moment in industry.
Okay, very clear. Hert?
I have a question related to the end of life of the bacteria. So, do they still produce plastics when they become waste? What do you do after the processing, after the generation of the plastic with the bacteria?
So the end of life of the bacteria is actually the end of their life. After we produce the bioplastics with the bacteria, we have to discard them because we really have to get the plastic out of the bacterial cell. So if that's your question, after one production, the bacteria, you have to cultivate new bacteria to produce new bioplastics.
And there is no risk that they keep on cultivating when they got in the free nature, for example?
No, after you get the bioplastic out of a bacterial cell, there's no possible way that they will keep being alive.
Okay, thank you.
Further questions for Hannelore? Lauren? Yes, go ahead.
Do you work together with industry in any way to see whether, for example, production costs are something that's attainable for them to do?
Yeah, that would actually be very interesting, of course. But my PhD research is a fundamental research, so I'm really looking at fundamental structure property relations is what I call them. So for me, it's not that important or not that useful to collaborate with industry. But I have some colleagues here at the VUB that actually do that. Yeah.
No further question. Maybe I have a very small one to to stop with. Where do you see yourself heading towards after your PhD?
That's fluctuating every day. One day I want to work in industry, the next day I want to become a professor, and the other day I want to work in science communication. So let's see what I will do afterwards.
A lot of possibilities lie ahead, that's for sure. Thanks a lot.
Thank you.
Another round of applause for Hannelore. We're going from bioplastics to epigenetics. Huang Yu-Xung might have some genius solutions for us all. Please give a warm round of applause for Huang Yu.
Millions of people are suffering from chronic, widespread pain, where even simple activities like walking or exercising can worsen their discomfort and pain. This is a harsh reality for those with chronic, widespread pain. A chronic pain condition where someone suffers from pain multiple areas of their body. But why does chronic pain happen? We try to explain chronic pain by looking at epigenetics. You can think of our genes as the musical notes on a musician's score, detailing how each part of our body should function. These musical notes need to be played at the right time and in the right order, which is where epigenetics comes in. Epigenetics act like a conductor for our genetic orchestra, controlling when each gene should be expressed or silenced. This precise direction helps maintain our body's harmony. When these genes work in harmony, they create beautiful, pain-free music. However, in chronic pain, it's as if the orchestra conductor loses the score, leading to a harsh mix-up of painful signals. My research focuses on these epigenetic disruptions, genes that are either overactive or underactive, and how we can correct them. One promising approach is called transcranial electrical brain stimulation. Yes, this non-invasive brain stimulation technique applies a safe, low-intensity electrical current to our scalp. Don't worry, it's totally safe. It will be able to correct the epigenetic errors, helping maintain genes properly, and reduce pain. With this, we can potentially slow down or even prevent the progression of chronic pain, allowing millions of patients enjoy their lives pain-free once more. Together, we are paving the way toward a brighter, pain-free future. Thank you.
Thank you so much, Huang Yu. Yes, Floor?
Hi, great page, congratulations.
Thank you.
Maybe this is taking us too far in the technical direction, but I was wondering, I was always thinking that working with genes is very complicated, very invasive. But I didn't know it was possible to work with genes or gene-related conditions through electrostimulation, so from the outside. Could you explain a little bit more about what the electric currents do to the genes, or how
Yeah.
it relieves the pain?
Yes, sure. Actually, in my research, we have two very promising pain-related genes. Yeah, it's called BDNF. Actually, the BDNF is very associated with chronic pain and also associated with the brain neuroplasticity. And so far, we know the TDCS, the brain stimulation technique, can control or manipulate the neuroplasticity. So we believe the TDCS reduce pain by modulating neuroplasticity. The mechanism behind that maybe is controlling the gene expression. Because before and after brain stimulation, the patients with chronic pain, we will find the BDNF protein expression changed. Yeah, especially for BDNF, after in patients with chronic pain compared with healthy people, the BDNF protein expression is too high. So we try to use brain stimulation to control, to bring the BDNF level back to normal, and then reduce pain. Thank you.
Another question, Leo or Lauren? You were first, actually.
Is the transcranial electrical brain stimulation like a one-off treatment, or does it need to be repeated with certain interval times, or how does it work for the patients?
Actually, for one session of transcranial electrical brain stimulation is usually about 20 minutes, and with 1 MA or 2 MA, it's very low, low intensity electrical current. Sometimes the patient even don't feel anything about their scalp. So they use this low intensity safe, okay, I need to focus, it's very safe, this safe electricity to help our patients to modulate or improve their brain function back to normal. Because as we know, in my research, we know, especially in patients with chronic pain, their brain function is, or I can say their brain excitability is too high. So we can use TDCS, use this small electrical current to slow down or back down the excitability.
And they only need one treatment?
It depends on which type of chronic pain condition.
Okay.
In my research, it's chronic widespread pain. So usually we need a long-term sessions, not only one. Yes.
We have still time for Leo's question.
Yes. So is the direct or indirect effect on your genes, meaning how is the excitability a result of difference in epigenetic regulation of BDNF, or is first the epigenetic deregulation and then excitability, and how do you take care of specificity? Because you have methylation, acetylation, ubiquitination. How can a TDCS cause specificity? There are actually two questions, sorry.
Okay. First, yes, the epigenetics contains different mechanisms. Actually, in my research, I focus on DNA methylation. Yeah, and then in our previous research by my supervisors, we find compared with healthy individuals, the DNA methylation of BDNF gene is a little bit lower. So lower DNA methylation means the gene expression is more high. So we have some solid foundation. We have already found some data set from patients with chronic widespread pain. So in my research, we will do it further to control it. Sorry for the second question.
No, how is it specific also for BDNF and not for all the other genes?
Actually, yes, we can say we cannot solve the pain problem by only targeting one gene. But we know BDNF, so far BDNF is so associated with pain. Not only the BDNF with pain patients, but also the BDNF associated with neuroplasticity, brain function. Yeah, and the brain function and neuroplasticity is so associated with brain stimulation. So it built a connection between epigenetics, BDNF, and brain stimulation and pain. Thank you.
I suggest you can talk further about it with Leo in the break. But thank you so much, Huang Yu.
Thank you.
Great job. Up next is Yara Saluena. She will teach us more about fungi. I myself really like fungi in a lot of different dishes, but she will not be standing in the kitchen. Really a warm applause for Yara.
Can you guess what the largest organism on Earth is? Maybe a whale? Perhaps a woodroot tree? Both these organisms look tiny compared to a honey fungus. You've heard right. The largest organism on Earth is a fungus. But how? I bet you've never seen a mushroom nearly as big as a tree. That's because mushrooms are just the visible part of a fungus. In fact, most of the fungus grows underground as a giant network, like the roots of a tree. This network, known as mycelium, can extend up to several kilometers in size, making it an abundant source of biomaterial. We can grow mycelium in the lab to produce sustainable materials for a circular economy. Mycelium feeds on waste. It requires minimal energy input, and it can grow into almost any shape. The materials are also lightweight, fire-resistant, and completely compostable. But then, why aren't mycelium materials everywhere already? In order to make things out of mycelium, we first need to learn how to produce enough of it in a consistent way. Different growing conditions lead to materials that have unique own characteristics, even at a microscale. In short, different incubation conditions, different growing conditions lead to affect the properties of the final material. My research focuses on finding the best conditions to produce more consistent and durable mycelium products. To do this, I start by growing the mycelium under different light and temperature conditions, and then I test their strength and performance of the materials. The materials like this, thanks to materials like this, your leather jacket could be grown instead of with animal harm. How about this? With materials like this, your home's insulation could be completely fireproof. With this knowledge, factories can produce uniform mycelium products for a variety of everyday uses. Help me in paving the way towards a future where innovation and sustainability go hand in hand, one mycelium jacket at a time.
Well done, well done. Questions from the jury? Yes, Arushi?
How durable are these products that are generated from the mycelium of fungi?
That's a very good question. These products so far show a high shelf stability. If they don't get wet, they can last for many years. We have produced materials in the lab that are still there for 4 years or something like that. And we expect that they can stay still for a long time, as long as they don't get wet.
Gert?
I have a question related to the lights that you are using.
Yes.
So I know that the spectrum of the light is important, but in some cases also, well, how it changes over time. So when first if you are working in a greenhouse, first this spectrum is important, and then when they're the flowers, it's another spectrum, and so on. Do you have the same issues? Do you have the same changes that you have over time for the light spectrum?
Indeed, that's precisely what I am mostly focusing on. I want to learn how the mycelium reacts in different light conditions, but not just different light wavelengths, but for example, different cyclings, and whether different, if the fungus is at a different stage, then it reacts different. So there are a lot of factors that I would like to tackle, and one of them is light wavelength, another one is light intensity, and a different one is light cycling.
Okay, thank you.
Yes, Marie-Laura, go ahead.
Thank you for your presentation. How expensive is this to make this mycelium material?
Yes, so having in mind that they can feed on waste, you can expect that the raw materials are not really as expensive. They also require minimal energy to grow, so as long as they have a temperature that is warm enough, they would reproduce and they would thrive there. And based on the light, based on the other conditions, it depends on what you experiment. But for now, producing this is a garage type of experiment.
It's a small one, Leo, then?
A tiny one.
Okay.
So you say a garage type of experiment, but how big of a garage do I need to make a leather jacket?
Yes. I would say that because this, which is similar to leather, it grows on surface fermentation. I would say that you would need as many surface as you would need, as fabric you would need for a leather jacket. But having in mind that instead of being fabric that you need like entire crops for it, like entire earth surface, you can stack them and grow them in shelf.
Thank you.
Okay, thanks a lot, Yara. Another round of applause, please. We're moving on to Morten Wilke. He will talk about ambiguity in financial markets. I would say, have a safe trip, Captain. Oh, you don't need it. Sorry. Go ahead.
In 2010, my mother lost her job. I was 14, and it was the first time that I heard about a financial market crisis. When I started studying economics some years later, I asked myself a very simple question based on that experience. How can I invest best in the financial market, knowing that it might crash, or more scientifically, knowing that I face uncertainty? And science suggests that there are two types of uncertainty: risk and ambiguity. Consider the following example. Imagine that you're a captain on a ship navigating to a small island in the open sea. And luckily, you brought a map with you, and it shows some dangers, such as reefs on your way. That is when you face some risk. But now imagine that during the next day you spill some coffee over that map, so that you're no longer able to see where those reefs lie ahead of you. Meaning that from now on, you have incomplete information about the risk ahead of you. And that is what we call ambiguity. In financial markets, of course, we don't have maps, but we have models to guide our investments. And the financial market crisis that made my mother lose her job showed that those models can be wrong. Most economic theories that are out there ignore this problem, but our research tackles it directly, using so-called smooth ambiguity preferences. What does this mean? It means that, coming back to our example of that ship, that you as a captain charter your course to that little island based on multiple guesses where those reefs lie ahead of you. By using those smooth ambiguity preferences, we help investors like you and me to optimally adapt our investment strategies to ambiguity. For example, we show under which circumstances it is optimal to invest in a stock market index, for example, such as the BEL 20 here in Belgium or the Dow Jones in the US. In a nutshell, we help investors to make better decisions. Or more loosely speaking, we help you to better navigate your ship through the uncharted sea of the financial market. Thank you very much.
really like the image. Thank you for that. Jury. Yes, Lauren.
I was very enthusiastic to go for the mic. Hi, Morten. Are investors the biggest winners or people who have something to gain from your research in your eyes? Or are there other parties who have a stake?
Yes, so what is important to know about our research, we are focusing in our research on people that are private investors, so small investors. And maybe to counter a little bit the impression that it's more about making rich people more richer, it's actually about helping people to make better decisions in financial market. And it's especially important at this moment because, as you all know, that's my second strand of the literature, of research. The pension system, for example, is under pressure all around the world, and people are thinking about moving to the financial market to supplement state pensions. So I think it's really important, and that's also why all people, I think, benefit from our research, that we make better decisions in the end.
Marie-Laure, please go ahead.
Thank you for your presentation. Do you implement your research then via a tool, or how can you please explain how this works in practice?
So what we are basically doing, I'm myself invested in the financial market, and I invest accordingly to what I basically recommend. But it's not that I'm implementing myself basically the investment strategies that we're recommending, but yeah, it's basically also about explaining what we actually see out there, because the investment strategies that we're, for example, recommending are already implemented in the real life, and basically, yeah, so we can explain why people are doing this, why it is optimal indeed to do this.
Thank you.
Okay, more questions from our jury? Or I forgot to mention, also the audience can ask a question if they like.
Now I get the audience question. Yeah.
Yes, yes, yes. It's on its way.
I cannot hear anything.
Can you hear me?
Still not.
So my question is...
Yeah.
Yeah, okay. My question is, do you only count, or is your tool only for the individual win? For example, if I invest in oil, I'll have individual gains, but my community might suffer?
Very good question. Very, very good question. Actually, when I was sitting there, I was thinking maybe someone will ask about this. So we have an objective in our case, which is indeed that you maximize your individual profit. That is correct. But our tool, or what we are using, can also be applied to another objective. And actually, that's kind of a hot topic now in the research. For example, my supervisor in Germany, who's hopefully watching, she works on this, basically, on how to implement those theories to actually maximize the objective that you just mentioned.
Okay, very cool. Thank you, Morten. Another round of applause, please. So, from the uncharted seas of Morten, we're crossing the ocean towards Peru, where we meet up with alpacas and Dora Shikogu. Please, a round of applause. Here you go.
Don't we all just love alpacas? And most of you love alpacas because they're super cute. And it's true. But I love alpacas because they make a special type of antibodies that can help us fight cancer. Antibodies are proteins that our body makes to protect us from germs, diseases, infections. And people at the VUB discovered that alpaca make a special type of antibodies that can be used to make nanobodies. Yes, nanobodies. They are as effective as antibodies, but 10 times smaller. And this is their biggest strength. These tiny nanobodies can be injected into the bloodstream, and thanks to their small size, they can easily and quickly find hidden targets in the body, such as cancer cells. This way, in our lab, we use nanobodies to detect or attack cancer cells. Sounds amazing. Here is the catch, though. As effective as nanobodies are, they do tend to stick in the kidneys for too long. And this makes it challenging to track down cancer cells close to the kidneys, and even worse, it can damage the kidneys. This is where I come in. My PhD research is all about better understanding why do the nanobodies stay in the kidneys for so long. How do I do this? Well, by studying nanobody on 3 different levels. First, I study the behavior of nanobody in the entire body from the moment they are injected. Then, I focus on the kidney itself. And I track down the nanobody, and by using advanced cameras, I can reconstruct in 3 dimensions where the nanobody sticks. Finally, I zoom in even more and look for nanobody on a microscopic level in the cells of the kidneys. And by better understanding all of these 3 levels, I can then hopefully develop a nanobody that still tracks down cancer cells, but doesn't stick in the kidneys at all. So yes, we agree. Alpacas are super cute. And don't you love them even more, now that you know that they can help us fight cancer?
Great job, Nora.
Thank you.
Jury, please, go ahead. Leo?
Yeah, so Gert reaching for the microphone, but I was faster. Tell me more about these advanced cameras. What are they, and how high is the resolution? Is this super resolution cameras?
So it is not super resolution, because the goal of this imaging technique, this advanced camera, is to see the whole organ at once. So I have my whole kidney, and I can image it entirely and reconstruct in 3 dimensions. And if I want to have a higher resolution, and like I say, look in even deeper, then I use higher resolution to look at the cells of the kidneys. But so to answer your question, no, it's not high resolution, but you can see the whole kidney at once.
Okay, Gert, go ahead.
I have a question related to the alpacas.
Okay.
How is he or she, how are the alpacas solving this problem? Do they have a special mechanism to solve the kidney problem, as I could call it? Or
No, so they don't have a special mechanism to solve the kidney problem. That's our job, I would say. But what we can do is better understand which types of nanobodies stay in the kidneys longer, because we do see that some nanobodies don't stay in the kidney at all. Then we can try to focus on these nanobodies and these type of antibodies.
Another question by Lauren?
Yes, you talked about the 3 levels at which you look at nanobodies. Can you explain to me, perhaps with an example, why it's important to not just leave one of those levels out in your research? So there's a kidney problem, and the 3rd level concerns the kidney itself and looking more deeply, but why are the other levels important?
Well, because the 1st level, where we look at the entire body, is important because we want to know, is a nanobody targeting the cancer cells, because that's its main job? And then, is there accumulation somewhere else, maybe in the liver? So that's very important. I cannot only focus on the kidneys. Once I know that, I can go further to the other level. I look in the kidney, and like I said, I look at the whole organ. Where is the accumulation? Sometimes we see it's only in one layer of the kidney. Sometimes we see it's in the whole kidney. And then I look in the cells, because we see that the nanobody is sometimes in the cells and sometimes on top of the cells. So that I have a good overview on all the different levels where the nanobody goes.
Maybe a last question by the audience? No? Don't be shy. It must have been very clear, Dora. So, thank you very much. So, we know from research that it's quite unhealthy to sit for a very long time. right? So those who have been listening carefully to me in the beginning know what comes up. I will ask you to stand up and follow the instructions. Enjoy.
Hey allemaal, welkom bij een nieuwe Fitbooster. Ook vandaag heb ik weer 3 nieuwe oefeningen voor jullie voorbereid en we gaan samen ontdekken welke dat zijn. De eerste oefening gaan we de enkels losmaken. We gaan de handen in de zij plaatsen en de enkels van buiten naar binnen bewegen. Doe maar allemaal mee. Maak mooie rondjes en we gaan naar binnen bewegen. Super. Nu de andere enkel. Naar buiten bewegen, mooie rondjes maken. En nu naar binnen. Oké, nu dat de enkels los zijn, gaan we een stoel gebruiken om daarop een beetje squats te doen. Een squat, je plaatst je voor de stoel en we gaan telkens met een gestrekte rug naar beneden, de stoel een beetje tikken en terug naar omhoog. We gaan dit een paar keer doen. Een squat. En naar omhoog. Naar beneden. Naar omhoog. Nu gaan we 5 seconden aanhouden. 1, 2, 3, 4, 5. Naar omhoog. Laatste keer. 1, 2, 3, 4, 5. Heel goed. Nu dat we de hamstrings hebben gebruikt, gaan we ze ook stretchen. We plaatsen 1 been voor en beide handen op het andere been. We duwen op ons gesteund been, zodat de hamstrings van het andere been worden gestretcht. We houden dit even aan. En nu doen we de andere kant. Dus het gesteund been wordt ondersteund met de handen. Heel goed. Oké, alles losschudden. En dan zit het er weer op voor vandaag. Bedankt dat jullie erbij waren. En dan zie ik jullie tot de volgende keer. Dag!
You were all amazing. Thank you for that. Me, myself, I rather have some chocolate during a break. And hopefully Elisa will tell us more about how they can safeguard for us eating chocolate. A round of applause for Elisa van der Zanden.
This here is a bar of chocolate. I really like this, and I'm sure I'm not the only one here. For over 5,000 years, humans have savored the fruit of cacao trees. It began with ancient civilizations in South America drinking chocolate, and later Europeans started to plant cacao trees on other continents, and we started to produce and consume chocolate on a large scale. However, despite our long history with this tree, one fundamental mystery persists. Who are the pollinators responsible for the reproduction of this tree? Pollination is a process where the pollen of the male parts of one flower are being transported to the female parts of another flower. We know that in the case of cacao trees, this is done by tiny insects, and that they look like this one. Measuring just 1 millimeter, these cacao pollinators are incredibly small. Even under a microscope, they are almost indistinguishable from one another. This makes the identification process quite challenging. And to overcome this, I use a method called DNA barcoding. Imagine scanning the barcode of a product at the supermarket. It provides all the essential details: name, price, and origin. Well, DNA barcoding is similar. We extract DNA from an insect and compare a specific segment to a vast global database of insect DNA. Based on the results, we can identify and classify the insect species accurately. By applying this method to pollinators that we catch on cacao flowers, we can find out which species are involved in cacao pollination, even if they are too small to be distinguished visually. We can even find out where the pollinators breed by comparing their genetic barcodes to those of insects captured in different parts of the plantation. But, so you're just all people here, you like chocolate, so do I. How is it relevant to you what pollinates this tree? We get chocolate anyways, right? Well, you might have noticed recently that chocolate is becoming significantly more expensive. So it's in our best interest to make the production as efficient as possible, right? Well, a way to do that is by optimizing pollination. As currently only 1% of the flowers are being pollinated, there is still a lot of room for improvement. By discovering the secrets of cacao pollination, we hope to boost the production and to contribute to a sustainable future where we still get to enjoy this bar of chocolate.
Thank you very much, Lisa. Jury, you have questions for Elisa? Floor?
How new is your field of research or your type of research? Is it already being applied to other plant or tree species, and are there any promising results there?
So DNA barcoding in itself is not a very new technique. It's been around for quite a while. But the way in which we apply it to identify species that you can barely see on such a scale, because we go to several continents, that's quite new. And most plants, their pollinators are actually not that as small as sweet cacao. So the easiest way to just identify is, well, you catch them, you put them under a microscope, you see, oh, it's this one, and then you know what it is. But since they're so small and you cannot distinguish them visually, we need to find another way. And this works.
Arushi?
You said you go to different continents to look for these pollinators. Do you find any differences between different places? Is it the same species that is responsible for this pollination, or are there also differences in terms of regions where you find these in the species?
So far, we find that there's definitely not one species that pollinates cacao trees. It's a whole range of species, but they all belong to one family, Ceratopogonidae. Then we compared the different, so cacao is originally from South America, and on this continent we find a higher variety of species than we found in Malaysia last year. And this year I'm going to Ivory Coast, and I expect also to find there a lower diversity of species, because it's been introduced in these continents.
Leo?
So you sequence the species. Do you have to sequence the entire genome, or are you focusing on certain aspects of it?
We sequence one gene, mitochondrial gene CO1. For barcoding, that's enough. It's the reference gene in a database, and everyone who barcodes insects, they all sequence the same gene. And it allows you to separate different species from one another, but within a species, the variety is so small that you're able to see properly how many species you have and which one.
Okay. That's the last one.
Yes, so I have a question related to the cacao tree itself.
Okay.
So now you say it's 1%, only 1% of the flowers that, well, basically produce cacao. But what's the optimal one? Because I can imagine if you go to 10, 20, 30%, that, well, the fruits will become smaller as the tree is overloaded with fruit. Do you have any idea about what would be your optimal percentage?
So yeah, to have cacao fruit, we don't only need pollination. The tree also needs enough nutrients, light, and water. And so the pollination is a limiting factor. So by increasing pollination, you will always have more production. But of course, when you come to the maximum capacity of the tree is going to abort more fruit. And right now, from what we have seen, the optimum is about 2 to 3 times as more than what's naturally happening. So you could double or triple the production if you optimize pollination.
Thank you.
Thank you very much, Elisa. I was told the next finalist has superpowers, but did he bring escape or will he keep in disguise? Let's see.
Thank you.
Frederik van Hassel. There you go.
Well, no spandex and capes, unfortunately. Yeah, secret identity and all, you understand. I vividly remember my very first night shift as a brain surgeon. The first patient I ever had came in. This is him. He looks a bit like a Tony, don't you think? Let's go with that, Tony. Tony came in and he had a trauma to his head. Right? The pressure inside his head was increasing rapidly, and he wouldn't survive if we didn't act quickly. So I had to place a life-saving drain in the middle of Tony's brain. Now this can be a challenge. Targeting a point in the middle of the skull is a bit like shooting an arrow into a dark room and hoping to hit a target of about 10 centimeters wide, hanging somewhere in the middle. So this is me imagining my target. I knew perfectly what I had to do, but still, if only there was some way for me to look inside, have some futuristic superpower, some X-ray vision. I would close my eyes, open them, and I would see exactly where I need to be. Well, thanks to my research on the use of augmented reality in surgery, this is now no longer science fiction. So what is it that augmented reality does that allows us to do this? Well, augmented reality, like virtual reality, augmented reality shows you digital images in front of your eyes, with the key difference that with augmented reality, you can still see your surroundings, like the patient, like Tony. Not that important for surgery, I'd say. Using special glasses, like the Microsoft HoloLens here, we can then reveal the patient's inner anatomy, certain waypoints and landmarks that we use for surgery. Think of it as a GPS that the surgeons can use to navigate through the human brain. But how did we do this? Well, first of all, we had to make sure that our GPS was fit for surgery. It needs to be immensely accurate, not down to a road or an address, but down to a nerve. So as soon as we ensured this millimetric precision, I tested it in the lab with already spectacular results. So next, off to the hospital. I implemented our augmented reality brain GPS in real-life cases, and for the better. We were able to place these drains inside the brain with more precision and less complications. But it didn't stop there. We were even able, using this technology, to improve the surgical plans that we use for brain tumor surgery, like the one you see here, with the tumor in green. All because the surgeon could see exactly where he needs to be. Thanks to my research, brain surgeons like myself are now able to wield superpowers, to see the invisible and do the impossible. Thanks.
Thank you very much, Frederik. Yes, jury. Lauren?
Hi, Frederik. Can you explain a little bit what other disciplines you are working with in the research team and what their role is compared to yours?
Absolutely. We're working mainly with disciplines that allow rigid structures to be tracked. Because, of course, if you have intestines and you use the images from beforehand, the scans, the MRIs, the CTs, those create your 3D model. But of course, intestines, they will be moving about during surgery, so it's very difficult to keep track of them. So we use rigid structures to be tracking anatomical parts of the patient. So what we use, neurosurgery helps, spine, ortho, and maxillofacial surgery are the ones that have progressed most. But there's been research on all medical disciplines that has been going on for quite a few years.
Okay, that's clear. Another question? Hert, go ahead.
Well, I have a question related to how you get the accuracy. Do you have reference points on that you have to put on the head of the patient, or can you comment on that?
Yes, absolutely. Actually, what we have been using for quite a while are infrared reflective markers. I'm not going to take all the credit for the tracking accuracy. This is all thanks to the engineers from the VUB who were able to develop this with a tracking accuracy of about 0.7 millimeters. And these spheres we attach to the head of the patient or any other part that you want to be tracking. And these can be recognized by the headset very easily. And the unique pattern of these spheres allows the headset to know, okay, this is exactly where the head, for example, of the patient is positioned.
Okay. Other questions? from the jury or from the audience? Yes, Floor, go ahead.
I'm going to ask a question like a child because I'm not sure if I understand correctly. So does the augmented reality is based on the information that you have on the patient?
Correct.
So what happens if someone comes in like this, like it's really an emergency, you don't know anything, you still have to, maybe the medical file isn't uploaded yet. How does that work exactly?
So we use the imaging, the preoperative imaging. Of course, for a tumor case, this is done a bit in advance and the changes are not that instant. For a trauma case that comes in, you have a hemorrhage or you have some swelling somewhere, it needs a drain. Patient will always undergo a CT scan to have the diagnosis. And that's what we're going to use to create a 3D model instantaneously, which is then within 5 to 10 minutes in the headset, and we can use it immediately after in the emergency room or intensive care unit.
So you can use it in emergencies, like the
Exactly.
specific instances, and for longer treatments of diseases.
Absolutely.
Okay, thank you.
And if anyone is interested, you can always try it out later. Feel free. Just come up to me, have it with me. So.
During the drinks, let's say.
Yeah.
Okay, thank you, Frederik. Okay, we arrived at our second last speaker already. While Wang Yu compared epigenetics with music, Silke Marinissen is actually, in her research, using music among prisoners. I would say, take it away.
You're in a very small cell of 10 square meters for 23 hours a day, and you share this room with a stranger. How would you feel? For many people in prison, this is their everyday reality, and 99% of them will eventually return to society. So it's crucial that they are well prepared for their return. And one possible way to do so is by making music. I can hear some of you thinking, did she just say music? Well, yes, music is a powerful tool for people in prison. Sometimes they can sing in a choir, craft beats in a hip-hop workshop, or they play in a band. And this not only teaches them how to sing or how to play an instrument, but it also helps them with their self-development. They become more confident, and it gives them a chance to share their emotions. However, music activities vary widely across prisons. In some prisons, people cannot make music whatsoever. And to make music accessible in every prison and for everyone who wants to make music, my research uncovers how, why, and under which circumstances music activities in prison work best. And I found that it's important that the created music should not stop within the prison walls. People often write their own songs, and during my observations, I have heard inspiring songs about losing a family member, what it's like being homeless, living on the streets, or the harsh reality of living inside of prison. And when people can share these songs with the outside world, they feel connected again. And what's in it for the society? Well, we can learn so much about the valuable life lessons within these songs. They shed light on societal issues, such as, for instance, homelessness. And this might be the reason why people ended up in prison in the first place. And that is why this is my favorite music album. This CD is made by a band called Cellar. And Cellar exists out of 8 people who live in the prison of Leuven. And their music already taught me a lot about the prison, but also about society. Thank you.
Thank you so much, Silke. Well done. Jury. Some questions for Silke. Leo?
Yes.
Yeah.
how easy is it to have prisoners, you know, go into music, or are they already inclined to play music, playing an instrument, sing songs, write music? Because else it might not be the easiest thing to do. It's easier to work out, you know, push some kilos.
Yeah, it's true. So of course, maybe there might be people in prison that are not interested in making music and rather go to the fitness or something like that. But there are also people that really want to play music, and some people already write songs or lyrics in their cell already, and they really want to make music. And then if there is an activity in prison, they can go to it. But most of the time, these activities only happen for a very short time, for instance, 5 sessions, which are 3-hour sessions, and then that's it after that. So, yeah.
Prisons do not have an orchestra or a choir or stuff, no?
There are some prisons that do, but it's very, very limited. For instance, in the prison of Leuven, there is a choir, and they also have a music room where bands can go to rehearse. But the music room in Leuven is the only prison in Belgium or in Flanders that has a music room.
Okay, Hert, yes?
Well, I can imagine when you are in a cell together with somebody that plays music, but it's really not your style, that it's not helping. Maybe for him, but not for you. So what can we do about that?
I think that's a great question, thank you. It's true, when you're in a cell and you're sharing your cell and you're playing music and the other person doesn't like it, it might be frustrating. And that's the reason why it's so important to have these type of music rooms where people can go to and rehearse and have a space where they can make music, play their own genre that they like.
Okay, Lauren was first. Yeah.
I'm quite curious at how you do your research. So do you send out surveys? Do you talk face to face? Who do you talk to? And how would it differ?
Yeah. So actually I do a lot of stuff to get my results. I've started with talking to the people that facilitate these activities, so a music facilitator or a coordinator. But I've also done observations. I went to a music band rehearsals. I went to a hip hop workshop, and there I talked to the people who were there, so people in prison, but also the music facilitator. But also I did interviews with people in prison themselves to listen to their stories and how this has an effect on them as well.
Okay. Floor?
I had exactly the same question.
Great. Maybe a question from the audience? Or I also have one for you. I was wondering why you chose this topic.
Okay. I have been playing music since the age of 6. I'm an amateur musician myself, so it's something that I have experienced as powerful. Mm-hmm. And on the other side, prisons and the prison system is something that is very unknown and something that intrigued me already for a long time. So combining both my passion and my research interests is how I came on this topic of music in prisons.
Great story. Thank you for that.
Thank you.
Another round of applause for Silke. Yes, and then the last pitch of today will be given by Yuheng Zhao. And he's focusing on an illness that affects more than 1 in 10 persons. Please give a warm round of applause. Yuheng, the floor is yours.
Okay, so take a look into this bottle in my hand. You see stones in it. These are the stones that I picked randomly in the campus today. But in some cases, the similar formation could also be found in someone's kidney. And they are called kidney stones. Kidney stone is a disease which can cause sharp pain and even kidney failure when it blocks the flow of the urine. I know it very well because I was also an ex-patient since the age of 4. I remember that I went to hospital every week. I couldn't hang out with my friends. It's painful for a little kid. And nowadays, this disease is still affecting 10% of the world's population. But fear not, the little patient has become a chemist, and my research will help to understand this disease better. Kidney stones are made of tiny building blocks, mainly calcium minerals. These minerals are 100,000 times tinier than a hair, and this is a problem. Since they are too small to be seen, it is difficult to diagnose and treat kidney stones at an early stage. So most patients have already suffered a lot before they get treated. Therefore, I am eager to find out what happened at the beginning of the formation of these minerals that will build up to kidney stones. Now you may wonder, how do I study these tiny minerals that I cannot see with my eyes? Well, I decided to work with super smart computers. I can create 3D models of these minerals with software and mimic the environment of the human body. This allows me to watch how these building blocks behave in your body, just like watching a movie on the screen. To study what are kidney stones made of, we shine a special light on them. Different kinds of minerals will reply with different signals. By providing this information to doctors, they can determine the type of stones their patients have and provide a personalized treatment. And the best part? The treatment can now start at an early stage, avoiding unnecessary pains for the patients. With my research, we're paving the way towards a future where kidney stone diagnosis is not only precise, but also preemptive, ensuring a healthier and less painful life for millions. Thank you.
Wow, ending with a bang. Thank you, Yuheng. Questions from our jury members? They are still writing up. Leo, you can go ahead.
Yes, very nice. I was wondering, actually 2 questions. You said there's many different kind of kidney stones, yes, depending on the minerals. And so I assume that you first have to know that you have kidney stones, so you feel already discomfort, I guess, before it becomes painful. And second is, can you predict whether somebody gets kidney stones?
Okay. In fact, in the medication examination, if you have some tiny formations in your kidney, at the beginning you won't feel it. You won't feel anything until it grows like a very big one, and you will feel sharp pain or even blood in the urine. You will see it very clearly. But if you do your body examination very frequently, the doctors can see the change, for example, the change of proteins, the change of blood cells in the urine. The concentration will change when these kind of tiny formations occur in the kidney, even you don't feel it. Yeah.
Thank you.
Does your research also prevent kidney stones?
Yes, I think so, because at the beginning of the formation of these kidney stones, you may have, for example, different kinds of composition, for example. And these different compositions will require different medications to work better on them. So it is also one of my research topics that I will test some different medical medications, chemicals, on different shape or different structures of these tiny formations, and to see if they work well or don't work at all.
Yes, Lauren, go ahead.
So I have maybe 2 tiny questions. So am I right that you're doing both the computer modeling and testing things in the lab?
No, I'm
Sorry.
only doing the computational work, but we have
Okay.
collaborators in experimental chemistry and also in hospitals.
That's clear. And then a follow-up, how difficult is it to get the parameters right for your model?
Well, which kinds of parameters you mean?
Well, I have no clue. Can you
Okay.
explain me a bit more about the parameters and how difficult it is to get everything right?
Yeah, okay, I see it. Okay, so the kidney stones have already been studied a lot, and the doctors have already found that there are some chemicals or some compositions which will be predominant. And especially at the starting point when they began to form, you have mainly calcium phosphate and calcium oxalate, which will be present. And also, we have a very detailed database about these kind of structures, and we could have different substitutions, means that some part of the structures will be replaced by other chemicals or other group of structures. And we know that, for example, the carbonate, some kind of carbonate, this formation could replace it in some places. So we have this experimental database, and we can construct our model based on it, and we can track the behavior, the change in this development of kidney stones. Yeah.
See a question from the audience. I don't actually. Yeah, Floor has another one.
It's only laterally related to your research, I think, but am I correct? I didn't know that having kidney stones or having regular kidney stones is a disease. Do I understand this correctly? Because my dad has them and my sister has had them, and this has happened only once, and that was it.
No,
But do I understand that there are people who suffer from kidney stones regularly then?
Yes, yes,
Yeah.
because kidneys, the formation of kidney stones are highly related to your living condition, your diet, for example. For most patients, they have a very high occurrence rate because, for example, you don't change your lifestyle, you still eat the same thing as before, and you will have this kidney stone occurrence again and again. Yeah.
Okay, that's clear.
Thank you.
Another round of applause for you, Henk. Thank you. Yeah, you got it. So, the stress is off, but what happens now? A few announcements I have to make before you can go and grab a drink. For those sitting here, you will be able to vote on paper. When you arrived at the registration desk, you should all have received a little piece of paper. You can tick one name only. And then you can enter it in this box. Brigitte will go along to collect them in a few minutes. For those watching from home, you can also vote. You can send a name to rtdo@vub.be. Only those who registered before noon will be able. Well, only those will be collected. The other ones not. Just in order to be on the safe side, that we're actually collecting votes from people who actually saw all the presentations, of course. So you only have 5 minutes times to vote for those watching from home, so be quick about it. And then for all of you, we have a 15-minute break. So you can already go to the bar and have a drink and then come back and watch who the winners of the awards will be. My colleagues will let you know when to come back. So yeah, enjoy and see you back in a bit. Thank you. Welcome back, everyone. The moment has arrived. We're going to start off with the award for Best Research Summary. And I would like to ask Sven to join me on the stage and tell us who the winner is.
Hi. Hello, everyone. So today you saw 9 presentations, part of this Doctoral Derby finals, but you have to know that the trajectory started by writing a compelling and accessible research summary in not more than 200 words, so that's not a lot. My main message for the participants during the training was writing is deleting, keep it short. And I will also keep it short because I can announce that the winner of the Best Research Summary of the Doctoral Derby 2024 is Yara. Congratulations. You may come on stage. Here you go, that's your prize. So maybe you can tell a few words. How did you expect a prize? How did you write your text? Was it difficult? What did you learn? Very briefly.
Yes, well, thank you so much for this prize. I am very glad to have it, and I thank to my team in the VUB, the Mycelium team, and also everyone who has helped me in this outreach trajectory. So yeah, thank you. Thanks a lot.
Okay. So let it sink in for a while, and we projected the research summary of Yara, so let's be silent for a few seconds, and we want all of you to read this beautiful summary.
Yeah, it's there. Sven, did you also wanted to mention the runner-up?
Yeah. So it was a very, very tight game, actually. So a jury different than this jury deliberated on the text based on a number of criteria, which are basically the same as your pitch today. But I also want to give special credits to Elisa, because it was really, really, really tight. But Yara won with half of a point difference. Yes, thank you.
Then we go on with the audience awards. You voted massively, so not only those here, but there are over 60 votes online as well. And it was not a close call. It was quite clear who the audience thought deserved this prize. So I would like to raise a big applause for Dora Shikogu. Please join us on stage. Congratulations. How did you enjoy this trajectory?
Well, as I told my colleagues, it was very challenging because it's so different than what we do during our PhD. But I really enjoyed it and I learned so much.
That's great to hear. Thank you so much. You can join Yara over there, and then I would like you can join her there, and we'll come back to you in a few minutes. Then I would like to give the stage to Lauren, who will talk about how the jury found all the pitches and how they came to a conclusion.
We came to a conclusion, a lot of hard deliberation and one sticky note in the end. So it was actually, we discussed for quite a long time because really we were impressed by many of you, and it was quite evenly matched for us. The person we chose, the contestant we in the end decided that should have the prize, we all had the contestant in our tops on our short list, so each and every one of us. They shone kind of across the board, so it was clear what their scientific project was, what the societal impact was, and most of all, the story was really simple and accessible, and there was a nice balance of enthusiasm and calm, and I need to follow Sven's training because this is going on for quite a while. So congratulations to Hannelore Gerard.
Tell me, what do you think?
I'm happy.
Great. So how do you look back at the trajectory?
Well, it was quite intense, I must say. A lot of trainings, a lot of sessions, a lot of revisions, especially for this pitch. But it was really useful because I really think that it helped me to optimize my pitch. And I also really liked talking together to the other participants and thinking about each other's pitch. And it was really nice. Yeah, I really liked it.
Great. I would like to ask everybody to come here. Yeah. I was wondering, what do you think was the main challenge, or what was really, if you look back at it, that challenged you the most? And maybe going to start with you, Yara.
Well, I would say that fitting everything in 200 words or 3 minutes, it was quite a challenge. Especially because with a 3-minute speech, you don't really have the margin to be able to maneuver through the presentation. So it plays a lot of learning by heart, and I found that quite challenging.
For me, I think it's to keep it simple, because when you do a PhD, you think some things are simple for you, for your research group, but not for everyone. So to just reset everything and think, okay, what do people know and how can I keep it simple? I think that was the most challenging. Yes, indeed.
And for you, Hannelore?
I think it was to have one clear story, because you always have you want to explain like the general problem in society, and you want to explain the problem that you're tackling with your research, and bridging all of those parts in a pitch was most challenging, I think.
Okay. Was this the first outreach experience for you?
No, I really like participating in events like this and stuff. For example, the picture that was there, someone asked me like it looked professional, but it was from the science show that we did with the Faculty of Engineering, and there I also got training for presenting. So.
Okay. And for you, Yara?
I had been in a children's class before.
Okay. Also challenging.
And I volunteered for the microbiology microorganism day. But that's not a lot of research of outreach experience, I would say.
Okay. And for you, Dora?
I participated to Kinderuniversiteit. Yeah. So there I learned how to keep it very simple. Yes, but with colleagues.
Okay. Yeah. And then maybe a last question, just because I'm curious. What are you going to do with the 500 euros?
I'm still thinking about it. I can use it to do some imaging with advanced cameras because it's very expensive.
Yeah, sure. Yeah. Okay. For you?
Maybe I can use this money to improve the setup that I have for testing the conditions on mycelium growth.
I don't know if it's allowed, but I've for organizing workshops for children, I've already created like some setups to explain polymers and properties of polymers. And if it's allowed, I would like to expand that setup to organize workshops.
What do you think? I see Leo nodding. Yeah, that's perfectly fine. Another round of applause, please. Yeah, maybe just some final practical things. So you receive normally 2 vouchers for drinks, and you need in order to get your fries, but it's all you can eat. So enjoy, take as many as you like. Please return your cups back to the bar at the end. But enjoy and have a great evening.